Hello, everybody. I welcome you to the training for ATAC-Seq data analysis, or ATAC-Seq. I quite often will pronounce it ATAC-Seq, which is not correct. It's actually called ATAC-Seq data. Please bear my mispronunciation. ATAC-Seq or ATAC-Seq stands for Assay for Transposase Accessible Chromatin Using Sequencing, and that's basically the analysis of open chromatin regions in your genome to then predict regulatory elements like enhancer or silencer. And in this training material, we will present the kind of the quality control of all the data and how to predict open chromatin regions. Also, some visualizations and how or what you can infer from the results from this data analysis. Just for a short explanation, what kind of data we will analyze. This data comes from from this paper here. It is the first paper on ATAC-Seq methods, and it's a human cell line from CD4 plus T cells. However, the data is a bit too large. It comprises a few gigabytes of reads, and in order to now do a very quick training material or training, we downsampled, so randomly selected some of the reads or the read library, and we added also up to 200,000 reads from chromosome 22 in order later on to make or show you a better visualization. So we will focus mainly on chromosome 22 in this training material. Of course, this does not reflect completely real data, but we took it from real data. We will also compare the predicted open chromatin regions from this data to a positive control of a transcription factor called Ctcf. Ctcf is a well-known transcription factor and should correlate with some of our sites, which we will see at the end. And we will also, of course, look if you can find open chromatin regions in correlation to transcription start sites. In order to understand this training, you kind of already have to know how to operate in Galaxy. I will explain a few or will repeat a few steps which you get from the Galaxy introduction. And you also have to know a bit about quality control. Also there, I will repeat some of the things and we'll go through all of the plots. So we will use again Fastqc if you already know it. I will repeat some of the plots. However, I will not go that much into detail. I will go into detail for those plots which are important for ATAC-Seq. However, if you want to know more, then please go to the quality control training and to the Galaxy introduction to fully understand this training material. Okay, let's first start. So let's go into our Galaxy. First, of course, log into your account. And you can see I have already here on the right side data analysis open. Let's create a new history for our ATAC-Seq training. So click here on this plus icon, create new history. And let's name this training material like ATAC training. So with that, we can start to import some of the data we need. As I mentioned from this first ATAC-Seq paper, so we have here 2 read files, and we will also import this BED file from a well-known Ctcf binding sites. So let's first copy this. So click here on copy. And then we go here on the left side to upload data, where we then click here on this button, paste fetch data, and we simply paste these links in here. Of course, if you may know, you could paste each link individually and also assign already the data type, but let's do all three of them at once. And I will explain a bit again about renaming and stuff after the data is uploaded. Okay, let's click here on start. And then we have to probably wait a few seconds or minutes until this is done. Okay, let's make here a short break and come back if this is finished. Okay, let's come back. So the data is now in our history, as you can see. And the names are, a bit convoluted here now because I have not assigned it immediately. When I downloaded the data, I did not change the names or anything. So let's do it in the history itself. So click here on the pencil icon. And then you can already remove this whole information here just before you click here on save. Let's also go into data types. It says here interval. Let's first, because it's actually a BD file, let's change this to BED. And let's say change data type. Oh, I should have also should have saved the name. So let's wait until this is finished and we can change the name for these files. So let's remove this and click save. So this name is changed. And this whole information. So the names are a bit shorter. So this is now a BD file. And we can now again remove the information here, click here on save. And then we have these shorter names. And you can see, so this is now a BD file. And this is a Fastqc file. And this is also Fastqc file. So as you already can see, this ATAC-Seq data is in paired end, or was done in a paired end sequencing. So we have the first mate here and here the second mate. That's why you have R1 and R2. So this is one sample. If you, I repeat, if you don't know what I'm talking now about, so what's a BD file, what's a Fastqc file, and what is paired end sequencing, then please go back to the quality control. There we talk about Fastqc, paired end sequencing, and BD is covered also in other training materials. So apart from this data, we also need some other data as well. So let's scroll down here. So here you can again read up quickly about Fastqc and here about BD. We also will need some annotation later on for our open chromatin regions, as well as for the binding sites of the Ctcf, which we downloaded now. And in order to get this annotation, you have the tool in Galaxy called the Ucsc main table browser. The Ucsc table browser is well known if you need some annotations. And there is a very quick tool where you can directly import the data from Ucsc to Galaxy itself. So you don't have to go to Ucsc, download the data locally. You just go to this tool and import it into your Galaxy history, which is very handy. So if you to mention, if you are not questioning what I'm doing now, because you could actually, so I can click here on this button and it will immediately open the tool. Of course, if you want to search for it, you can also click copy paste the name and go into this field, search tools. Then you paste this name in here, and it will list everything which closely relates to your search terms. And then you can find, for example, the Ucsc table browser added in a moment. Here, the main table browser here. However, if you want to follow this a bit more, if you don't want to always switch and search and so on, so you can click here on this See Galaxy Training Materials button. I was already in the ATAC-Seq training material, but if you go into Epigenetics, under Epigenetics, you have the ATAC-Seq training. And if you then follow this training based on this button in Galaxy itself, then you can immediately click on these buttons here for these tools, and then it opens directly the tool itself, just so that you understand what I'm doing right now. Okay, therefore, we click here now on this button, Ucsc main table browser, or you search in the tool field for it. And then you get to this page where you simply have to click on execute, and then you are directed to the Ucsc table browser. And there we simply select for our clade the mammal, and we want the human genome, the assembly version from December 2013, HG38. And we want to have genes and gene predictions from GenCode version 36, where we want to have known genes, and the position, we simply want to have everything from chromosome 22, as I mentioned. As an output format, we select all fields from the selected table, and we send the output to Galaxy. And if you then click on Get Output, you then have to also click here on Send Query to Galaxy, and then the data will be directly sent into your Galaxy history. I don't do this now, I've already done it, because there was right now a little problem with Galaxy. But if you have done these steps, then you have this Ucsc data here in your history now. From this data here, if you would look into it, so click here on the View Data icon, then you see this is a GTF file listing you, or it's not, it's a table file listing you the genes from chromosome 22. And from this table, we want to have specific columns. So we bring it into a format which is readable for a certain tool in this training. So we have to process this data. And therefore, please go to the next step, where you have to click on this button here, or you search for cut columns from table. And there we copy paste this line here, the columns we want to select, and put this in here in cut columns. The table is tab delimited, as you saw, and we apply it to this file from Ucsc. And you simply click here on execute now. This Galaxy now produces the output. In the meantime, let me show you to organize this history a bit better throughout the training. You maybe know that you can assign tags to the data. If you don't know, then let me show you how to do it. You can click on a data set, and here is a button for edit dataset tags. If you click on it, and then it opens this tags field. And if you click on this button here again, then you can assign some text. And let's say this is the binding sites of Ctcf. So we give it hashtag Ctcf. And if you press enter, then this tag is assigned to your file here. And let's do this for the other files as well. So for the first mate of our ATAC-Seq data, we can click here again on edit dataset tags, here on the tag button, hashtag ATAC, and I don't know, right, underscore R1. So this is my first mate. And for the second mate, I do underscore R2. And if you follow the training material, then you see that we have to rename the data we have produced. So this cut on data form. So this result file here, we want to rename into chromosome 22 genes. So save this. And we also can give it a tag just to make it a bit more visually appealing. So hashtag chromosome 22 genes. And this will help you to later on see which results come from which input data. And now let's begin the actual training, so to say. You know from the Galaxy introduction and also from the Galaxy quality control training that in any next generation sequencing, experiment, and data analysis, you have to do a quality control to see if your experiments worked, if there was a quality breach with the sequencing, or with your experiment. And also to see if you have technical biases in there, which you assume and anticipate from your protocol. And to check this, we use a tool called Fastqc. So you can either search the tool in the search tool field or click here on this button. And we now apply Fastqc to only to our first mate as a demonstration. So select here a single data set, and then click in this field and select the R1 file. All other options we leave as a default, and we execute the tool. And then you already see, you can now, so the tag is from ATAC R1 is assigned to my result files from Fastqc. So I can immediately see that this Fastqc report comes from the first mate. So this might take a while, and we do a short break here. Okay, let's continue. We generated the Fastqc report for our first mate. And as you can see, so based on our tag, the tag for the tag ATAC R1 is assigned, and we know immediately that this Fastqc report corresponds to the input of the first mate. You know maybe from the Galaxy quality control training that Fastqc produces two outputs here. One is the raw data, where basically, well, it holds the raw data. And then you have the web page, so the report with the visualizations. And if you click here on the view data icon, then we get, or we are greeted with the basic statistics about our data. So we see the name, we see the sequencing device and version, we see how many sequences are in the read library. Then we see how many sequences are flagged as poor quality. What is the sequence length? So we have a constant length. So all of the reads have a constant length of 50 bases. And we have here the average GC content in percent. Then we see, based on the most important plot, the per base sequence quality, where we see on the x-axis the position in the reads, on the read library, and on the y-axis the Fred score, so the quality score. And we have per position distribution of this quality for the position. And we see in the blue line the average of the quality. Or we can say from our experiments here that it's very good. All of the positions are in the green area, so above 28. So nothing is in the red area, which is the visualization for very poor quality. If we scroll down, then we have the Pertile sequence quality, which stands for, which is shown if you do Illumina sequencing. If you would see a red stretch signaling a poor quality, that this is an indication that the sequencing did not work quite well. So there was a quality breach in the sequencing itself. This is kind of cold to hot encoding here. So blue colors means very good quality, red colors poor quality. And you see here the position in the read on the x-axis, on the y-axis the lane of the flow cell. Please go to the Galaxy quality control training if you don't know what this plot or what this plot actually means. So there are it is described more into detail, and also please look into the Fastqc documentation to understand better. But if you again see a big red stretch or a lot of red stretches here, it would signalize or it would imply that there was a problem with the sequence. And here it's pretty good for our data. We have here the per sequence quality scores, so the distribution for our reads on the x-axis, the Fred score, on the y-axis, the reads with that Fred score on average. And you can see a lot of sequences definitely above 28. So this plot correlates with the per base sequence quality. And it shows basically that our read library has a very good quality. Now we have here a warning. It's not a heavy quality breach. A heavy quality breach would be kind of a red stopping sign. We have here just warning, signaling that we have here nucleotide bias. So this is the per base sequence content. On the x-axis, you have the position in the read. On the y-axis, the fraction of the reads with that nucleotide at that position for T, C, A, and G. And you see in the beginning, we have a heavy nucleotide bias. This is anticipated for the ATAC-Seq data. Because of the transposase, the TN5, which is used, this can introduce, I mean, the newer protocols, the bias is more or was more and more reduced. But however, there is a bias anticipated, so don't worry if you see a warning here. This is what I would expect from ATAC-Seq data. Then we have the per sequence GC content. We have human data. So we anticipate kind of GC content of round about 50%, but lower than 50%. In blue, the theoretical distribution. In red, the empirical distribution, so the distribution of our actual data. On the y-axis, the reads with that GC content. And it's here a bit spiky, probably because we downsampled the read library, and we have, which we will see in a moment, still some adapters in our data, or potentially some adapters in our data. Then we have the per base end content. You can see there are no ends in our reads. So here's the position in the read on the x-axis, on the y-axis, the fraction of the reads with the n at that position. And you maybe know also from the quality control training that an n stands for, or it's a placeholder character if the base calling algorithm of the sequencing device does not assign an ATAC-Seq because the signal was not good enough, then it outputs an n at that position, signaling, okay, this position is very, very, we have not a high enough confidence that this is an ATAC-Seq, and therefore we have to put an n at that position. And here you can see none of the reads have in the positions an n, and therefore we have a very good quality. Sequence length distribution, as I mentioned, all of our reads have a sequence length of 50. And then we have here the sequence duplication level in red. If you would deduplicate, in blue the actual distribution. On the x-axis, you see the duplication level, and on the y-axis, the fraction of reads with that duplication level. And you can see we have here a peak. So we have a lot of sequences in our read library which are at least 10 times duplicated. And Pcr duplicates normally happen if you do Pcr. And the more, so the higher, or the more Pcr cycles you do, the higher the sequence duplication levels you have in your data. And this is always a question so based on this, for some NGS experiments, you do definitely a Pcr deduplication. However, for ATAC-Seq data, this question always depends on the experiment you do. So not immediately, don't do a deduplication always for ATAC-Seq data. You have to ask if it's really necessary and if it's really worth also to do a deduplication for your ATAC-Seq data. The last things in this Fastqc protocol to mention is the table for overrepresented sequences. You can see there is a high count for this sequence, and this is basically our adapters, which are still in the data. And you see that also in the adapter content plot. So here we have Nextera adapters still in our data, which is shown here, and therefore we have to do an adapter trimming. And now we want to remove these adapters. So let's go back to our training material, and you have here a mock-up of the Nextera read library. And you can see, based on the mate, we have different potential read or adapter sequences. So our first mate, which here starts at the 5', has a potential read-through here into the I7. And on the other mate, the R2, where we have here the 5', we have a read through here into the I5, And therefore, we have two different sequences for the R1 and the R2 that we have to remove. Furthermore, we also want to take care of low quality reads, so bases with low quality. So we also do a quality trimming. This is what you all quite often do for next generation sequencing data. And we want to remove reads which are too short. Normally, you set a cutoff, at least for ATAC-Seq data, and also for other NGS experiments like ClipSeq or ChipSeq, round about lower than 20 bases. We want to remove these reads because later on in the mapping, they are, of course, easier to map because the sequence is not as unique anymore, and it's therefore very easy to map the read and find the position in the genome. However, because the sequence is not that unique anymore, or becomes less unique the shorter it gets, you will have more of these reads mapped to multiple positions, and then you are unsure anymore what the correct position of the read in the genome is. And since we are interested of open chromatin regions, I mean, ATAC-Seq, we investigate these open chromatin regions. And we want to have exact positions. So it's crucial to have reads where we can say with a high confidence they come from this position, and this position is then signalizes as open chromatin regions. And therefore, we want to remove reads which map multiple times in our genome. And one cutoff is therefore the length. So let's first click here on the Cut Adapt button or search the tool in the search tool field. And then the first thing to do, we have to select here at the question single end or paired end. As I mentioned, we have paired end data. So we select here paired end. And then we select for first Fastqc file, number 1 here, the R1 file. And for second Fastqc file, so number 2, the R2 file. Now you can maybe ask, why do we have to select here paired end? Why not apply? I mean, I could also do single end and apply cut adapt twice, right? So one time for the R1 and one time for the R2. The answer for that is you have to keep the files synchronized. And as I already mentioned, we will do a filtering with cut adapt based on the length and the quality. And if one of the mates are removed, so let's say one read here in R1 is removed because it's too short or the quality is not good enough, then you still have the other mate in your R2 file. And then you have one less read in the R1 file in comparison to the R2 file. This will be a problem for the mapping because the mapper needs and paired ends, if you have paired end data, that you have a file which have the same number of reads in there. So if they are not synchronized, you will end up in an error if you do a mapping. And then either the mapper complains that there's something wrong with your format, or you get generally an error saying there's something wrong. And normally, or quite often, this has something to do that you miss or not use the adapter trimming tool correctly. And both read files are out of sync. So therefore, paired end, select both files, and now we have to say to cut adapt, look at these adapter sequences and try to find them in our read library. And therefore, So it's written down here in read number 1. So insert 3' end adapter. So click on this button. Then you select enter custom sequence. You can give here a label for the adapter, and we give it what's written here in the training material, Nextera R1. And now we give the actual adapter sequence. And we have this also here in the training material. So copy that string and paste it in here. So this is for the first mate. Now we want also to remove the adapter in the second mate. And therefore we go to, so this is read 1 options. So we go down here to read 2 options. And we also insert here a 3' adapters, enter custom sequence. Then we say here, Nextera R2. And we copy here the sequence for the adapter. And now we want to set the quality and length filter. So go in filter options. So we have here adapter options, and then we have filter options. And then we say here minimum length 20. And then we have in read modification options, quality cutoff, and here we say also 20. So we filter reads out which are lower than a quality of 20. And we also want to have an additional cut adapter report. So go into output options, and here you have report and set it to yes. And now we click on execute if I'm not forgot something. Okay, yes. So let's click on execute. And then you see already what I mentioned with the text. We clearly see here, okay, this cut adapt was applied to my 2 mates of my ATAC-Seq data. So this might take a few seconds, and therefore we do a short break again here. Okay, if cut adapt is finished, you will see that you have now 3 output files. 2 correspond to our trimmed reads now, so the first mate and second mate, and the third file is our cut adapt report. It gives you now an idea about general statistics about the adapter trimming. For example, we see here that for our first mate and our second mate, around about 14.5% of reads contained adapter sequences, and they were removed. We have also around about 0.7%, so 2,100 reads, which were filtered out because they were too short. And from 285, so around about 285,000, 283,000, so 99.3% of the reads passed the filter and are still in our data. So this is also always important to check, just to see if you have done something wrong with the adapter trimming. You have to definitely check also the adapter trimming still. I mean, the statistics is good, but to definitely check it, you have to apply Fastqc again. So let's search for Fastqc here and select multiple data sets and select the output files of cut adapt. So here for me, it's 11 and 12. For you, maybe different numbers, but definitely here read 1 and 2. And execute. In the meantime, I want to explain that we will, after that, apply the mapping, so an aligner. And we will use Pertile 2, which is a mapping tool which works quite well for ATAC-Seq data or ATAC-Seq data. There are three, so we have to optimize Bowtie2 for this data type, and there are three important things to mention. The first thing is that we will set it to -- very sensitive, so to an end-to-end alignment. That means we will use the entire read for the alignment. There's something called soft clipping. So there's also a mode which is different to this end-to-end alignment. Furthermore, we will map the reads to the canonical version of the human genome, so HG38. There exists some alternative or alternate load-C in the human genome. And in order to distinguish that reads map to these alternative load-C, because they would also map to the canonical chromosome, so they map multiple times. And in order to distinguish reads which map to these alternative load-Cs and reads which map to repetitive regions, and therefore also map multiple times, we have to map it to the canonical version of the human genome. The third thing, we will allow something called dovetailing. It's explained here in detail in the training material. But what it means is that CutAdapt only removes adapter sequences that are 3 bases long or longer. So if you have adapter sequences or bases, so 1 or 2 bases of the adapters in your reads, they will not be removed, and it can happen that one of the mates are a bit longer than the other. So here you can see this in the picture, that the mate 1 is a bit longer than the starting of the mate 2. And Bowtie would consider this as a non-concordant alignment and would remove it. However, you see this kind of event very often in ATAC-Seq data. And therefore, in order to keep a lot of our reads, we have to allow this dovetailing. Okay, that as a forward, let's check again our results for the adapter trimming. And let's check first for first mate, so the report, and let's go to adapter content, and you can see, okay, no overrepresented sequences anymore, and the adapter content is 0. Let's check for our second mate, and you see, yes, that as well. And here you can also check if your read files are synchronized again. So if you go into the basic statistics, you can see, okay, 283,155 reads in our second mate, and in our first mate, the same number. So we have the same number of reads in both files, and they're in sync. Okay, let's come to the mapping. Therefore, either search or click here on the Bowtie2 button. And as I mentioned, we have paired end data. So select here paired end, select the result files of our CutAdapt tool, and we select here for our Fastqc file number 1, the read, so the CutAdapt results for read 1, and here for Fastqc file number 2, the CutAdapt, the read number 2 file. Then, as I mentioned, we set it to -- very sensitive. Then we select use built-in genome index. And here we select search for the HG38 and the canonical version. And then we have to set or optimize it a bit further. So here you have the question, do you want to set paired end options? Yes. And we have here set the maximum fragment length for valid paired end alignments. And we set this here to 1,000. We know from this particular experiment that the fragment size is up to 1,000. So that's why we chose this number here. Obviously, maybe for your experiment, it's a bit differently. So don't just simply select here 1,000. Maybe for your experiment, you have to optimize this a bit differently. Then, as I mentioned, allow mate dovetailing, yes. And then we have set read group information. I have to find, do not set. Yeah, that's already done. Select analysis mode. This we have done, very sensitive. Do you want to tweak SAM-BAM options? No. And what we want still is some statistics about our mapping. So say under save the bow type mapping statistics to history, yes. Also always good to do. And then we execute the tool. So the mapping might take actually quite some time. So let's take a break here and come back in a moment. Okay, so hopefully the mapping is now finished. And let's take a look or a very brief look at the mapping statistics. So click on the eye icon for the file mapping stats for Bowtie 2. And then you can see, for example, that we have here around about 55% of our reads aligned concordantly exactly one time, and round about 43% aligned more than one time concordantly. Meaning if we would add this up, we have 97 to 98% of our reads aligned to our canonical version of our human genome, which is pretty good. Now, before we do the peak calling, which is basically the identification of significantly enriched regions, which then correspond to open chromatin regions, we have to do still some filtering. And we have to do a checkup. What we want to do first, and let's go back to the training material. Scroll down. And then we want to filter first read pairs with a certain quality, so mapping quality. And we set here the cutoff around 30. And they also have to be properly paired. So we definitely need to have one mate and the second mate together. And we want to filter out reads which map to the mitochondrial chromosome. Why? Because the mitochondrial chromosome is not that interesting to us. It contains a lot of open, or it's has no nucleosomes, and so the TN5, so this transposase inserts a lot of adapters in there, and therefore it's not of interest, this chromosome. So therefore we click here on the button or search for filter BAM datasets on a variety of attributes. And we select our alignment file from Bowtie 2. Here under BAM datasets to filter, we select the first filter, which is already in default, the mapping quality, and we say here it's bigger equal to 30. Then we insert another filter and say is proper pair, and select properly paired reads and say yes. And then we insert another one saying reference. And here we say exclamation mark chromosome M, which means it does not belong to chromosome or reads which map not to chromosome M, we keep, and all others we throw away, so which map to chromosome M. And that's basically it. So we execute, and then we probably have to wait a short moment. So let's stop. And let's come back. So this should be now finished. And based on just the file size, so let's see. So we have here 28.1 megabyte. And now after filtering, we have 50.2. So we actually filtered out half of the reads. If you want to include repetitive or reads which map to repetitive regions, then what you would need to do is adjust this cutoff for the mapping quality. And if you want to know more about this, so there is, I think, an interesting post about Pertile 2 about the mapping quality and mapping to repetitive regions. So if you search for it, you can find and read about this in more detail. Next, what we also want to remove are PCR duplicates. As I mentioned, we have PCR duplicates, and you saw this. You maybe remember from the Fastqc report. And these PCR duplicates are a bit of a problem because they would, if you would do a peak calling with PCR duplicates, they would give us or introduce a technical bias in our data and maybe produce them a lot of false positives. So a lot of regions which are signalized from our peak caller as open chromatin regions, but they're actually not open chromatin regions just because we have PCR duplicates in our data. And to remove that, we take this or use this tool called mark duplicates. And so we click here on mark duplicates or search for the tool. And then we select the BAM file from this filtering. And then we have to set exactly here this option, this long text. So I will just quickly search for it because I sometimes miss this option. It's actually right in the beginning. Okay, it says here, if true, do not write duplicates to the output file instead of writing them with appropriate flags set. It's a long sentence and we have to set it to yes, but this just means that we filter out PCR duplicates. So PCR duplicates are not they are not written into the in our result file, they are thrown out. And with that, we can execute the tool. And then we have to wait again. So I do a short break. So if that is finished, you have now two results from this mark duplicates tool. One file corresponds to some statistics. The other file is our BAM file where we have filtered out now the PCR duplicates. If you look into the statistics, then you have here a table giving you some information like, okay, we have investigated around about 135 or 136,000 reads. And from these reads, we have removed 3,000 or 3,600 reads because they are potential PCR duplicates. This is a bit hard to read, I can imagine. So in the training material, if you go under the section of mark duplicates, there is a tip formatting the marked duplicates metrics for readability. So where you can apply 2 tools to make this a bit more readable if you want to. However, let's skip that and let's come to one of the last points before the peak calling. And this is, we have to check the insert size for now our processed data. And this is very important for ATAC-Seq data because it has a very specific pattern you have to check. And if you see this pattern, then you're certain your ATAC-Seq experiment worked. And if it doesn't show, or if it's a bit off this pattern, then this is some information about some very heavy noise or a very big quality breach. And for that, we will apply a tool called paired end histogram. So search for this tool or click on the button here. And then we choose the marked duplicates, the result, the BAM file. We set we set as a lower limit 0 and an upper limit of a fragment size of 1,000, which we know and already used for Bowtie 2. And then we simply click on execute. And this runs now. I mean, I already know the results, and you can see already the results and the training material. So what you will get is this pattern here. And you can imagine you have now 2 mates. So one read, so they are 1 and they are 2. And there are different events or different pairs, so to say, for ATAC-Seq data. There is a potential of 2 pairs which are included in one open chromatin region. So no nucleosome in between. Then there is an event where 2 mates, so R1 and R2, actually surround exactly 1 nucleosome. And then there are 2 mates which surround exactly 2 nucleosomes and so on. And this you can see now in this insert size metric. So you have here on the x-axis the insert size in bases, and here on the y-axis the frequency, so the number of read pairs which have exactly this insert size. And you should see a very high peak, round about 50 bases, which corresponds exactly to these read pairs which are open chromatin regions and no nucleosome is in between. Then you have a smaller peak around 200 or smaller than 200 bases, which corresponds exactly to 1 nucleosome is in the middle of this read pair. And then you have another smaller peak around 400, which are 2 nucleosomes, and you can see the pattern. So then there's another peak around 600 from 3 nucleosomes and so on. And if you don't see this pattern, then something might went wrong with your data. So for example, if you have no nucleosomes, so generally a genome which has no nucleosomes, like the mitochondrial chromosome, then you see something like this here. So there's no bigger bumps, bigger peaks in there. If you have a very noisy ATAC-Seq experiment, then you see something like this. So it's very blurred, these peaks, and you see that the bump in 200 is very low. If you have a very bad or failed ATAC-Seq experiment, then you don't see these bumps at all. So then you have only here around 50, and then it's like no bumps. I mean, you can very vaguely see maybe here it's very slightly bumped in 200, but it's barely visible. And this totally failed. And again, so this is what we see, these very distinct peaks for 50, 200, 400, 600, and so on. So this is a very good ATAC-Seq experiment. And before we now go into the peak calling, let's do a bigger break. Okay, so let's come back to one of the biggest tasks now, or the most crucial task, the peak calling. We're left with that we have checked this typical insert size pattern for ATAC-Seq data and showed, okay, to see if our experiment worked, our ATAC-Seq experiment. And now we have to identify significantly enriched regions where we can infer these correspond to open chromatin regions. And two tools you can use for that is GenRich and Max2. Both were actually designed for ChIP-seq data, but they were also now optimized for ATAC-Seq data. It is always good to, so we will use here Max2, but it always is good to check another peak caller like GenRich to see if the results you get from a peak caller correspond to the results you anticipate, and also to check if some peaks are more robust. So if meaning that they pop up in one, not only in one tool, but also in the results of a second tool. So I highly recommend not only to rely on the results on one peak caller, but to use a second peak caller to check these results as well, because they have their false positives as well as their false negatives, since they have different models, so meaning different algorithms and data assumptions. Again, we will use here Max2, and we will optimize Max2 a bit for our data, meaning that we will recenter the reads on the 5' sites. Since we are interested on open chromatin regions, we have to recenter it so that we pick exactly that region and not pick the region which is exactly on the nucleosome. You might remember, and you see this in this picture, that the paired end data now encompass different events, meaning you have paired end or pairs which surround only open chromatins regions, or they surround one nucleosome, or they surround two nucleosomes, and so on. Max2 in its standard mode, since it was optimized for ChIP-seq data, it will recenter these pairs exactly on the center of the nucleosome, if there is one, of course. However, we do not want this behavior. We don't want to have a pile up at the nucleosome, right? Since it is closed chromatin. We want to have the pile ups in open chromatin, and therefore we will recenter our reads, our pairs, exactly at the 5' site where the TN5 binds and inserts our adapters. Furthermore, we want to take all of the reads into account, since all of the reads correspond to an open chromatin site. Max2, again, in its default mode, took both mates, the first mate and the second mate, recenters this for ChIP-seq data to have a pile up exactly where the protein binds. This we do not want. We want to have each individual mate that corresponds to open chromatin taken into consideration. So these are the 2 points I have to mention here. We have to optimize Max2 for ATAC-Seq data. Again, first, we have to recenter the mates, or we want to recenter the mates on the 5' ends. And then we want to take each mate into consideration for the peak calling. And this we will now do. And in order to do, and this we will do, and therefore we have to first apply BED tools BAM to BED converter. So we will convert the BAM files we have from the Mark Duplicates tool into BED, into a BED format. Else so we kind of trick now Max2 into believing that our paired end data is actually single end data. And to do this, we have to apply this tool first. So let's click here on BED tools BAM to BED converter, or search for it in the tools search field. And it did not run properly, so let's apply it again. And we apply it now to our Mark duplicates here on our BAM file. I think that's already it. Yeah, we apply it to, so just apply it to the data and click on execute. So we wait until this is finished. And I want to mention also, in the meantime, we only have here ATAC-Seq data taken into account for the peak calling. Of course, Max2 can incorporate control data. Since we don't have control data here, we will not do it. But for you, for your interest, Max2 is able to incorporate control data and GenRich as well. So I hope now this is not finished. Let's rerun. Not yet. Okay, in the meantime, I explain again this whole recenter. So you can see, so here's a short mock-up about what we will do. So this is the actual read, and we are interested on the 5' side. And we want to recenter the pi on this 5' side for the first mate and as well on the second mate. And therefore, we will shift the read 100 base pairs for the first mate to the left, for the second mate to the right. And then we'll extend 200 base pairs here to the middle. So we are left with this big stretch, so to say, at the end for all of our reads. Why do we extend it now for so much, for 200 base pairs? Explanation for that is if you would just take the 5' side itself, so just as one nucleotide, the data is too sparse to do the peak calling. So that's why we extend 200 nucleotides so that we have a bigger pile up or bigger distribution, so to say, and therefore to have a better peak calling. Now the conversion is complete and we can apply now Max2. So let's click here on Max2 call peak or search for it in the tools. And so now we have to again optimize the tool. So are you pooling treatment files? No. Chip-seq treatment files, so it says here chip-seq. Again, Max2 was designed for chip-seq data, but we can use it for other ATAC-Seq data with the right settings. So we apply it to this Mark duplicates. Pay attention to that it's this SBD file. Do you have a control file? No, we don't. Format of input files is single end BED. Effective genome size, we have, since the data comes from Homo sapiens, so from human, we apply it to Homo sapiens, so we leave it in the default mode. Build mode, so we have to now, exactly, so this is kind of now the build mode for chip-seq data. So this we don't want. We want to optimize it for other ATAC-Seq data. And therefore, we exactly set the extension size to 200. So this is, we will extend it to 200 and set the shift size to -100. So we shift the read now, what we have mentioned, to the left for the first mate and to the right for the second mate to 100 nucleotides. And then we want to have additional outputs. We want the peak as a tabular file. We want the peak summits. And we want the scores in BED graph file, in a BED graph file. And in advanced options, so click here, advanced options, we have to check another setting, composed, so this option here, composite, broad regions, no broad regions, use a more sophisticated method, yes. It's, if you want to know a bit more about this option, then please read the paper about MAX2. It would go a bit too much into detail now, but this basically gives us better results. And how many duplicated takes at the exact same locations are allowed. And we say here all. So how many duplicated takes at this exact same location are allowed? All. Since we have removed duplicates, we already know that there are no PCR duplicates in our data anymore, and we want to allow every read. It can happen that MAX2 removes reads based on just the exact same position. A read is mapping. Since we already checked for this, we don't want to remove these reads, and therefore we say, okay, take all reads we have now into take them into consideration. And with that, we can execute the tool. And this might now take a while, and therefore we do a short break here. Okay, let's come back. So the peak calling should be finished now. And now we want to do, for most of the people, the most fun part, and this is the visualization. For all NGX experiments, at the end, you will do the bigger analysis. And here we want to check if our ATAC-Seq peaks correspond to what we would anticipate. And for that reason, we downloaded, as you may remember, binding sites, a file which corresponds to binding sites of a transcription factor called Ctcf. So this is our positive control now. We want to see if the peak calling gave us now binding sites, so open chromatin sites that correspond to transcription factor binding sites of Ctcf, which is something we would kind of anticipate. And furthermore, we will also want to check later on if transcription start sites are also more covered, so have a bigger coverage, read coverage in our ATAC-Seq data than other regions, which we also kind of would anticipate from ATAC-Seq data. And in order to do the analysis now and also the visualization, we have to do a few steps to apply our visualization tools. First of all, we want to select only the binding sites of Ctcf, which corresponds to binding sites from chromosome 22, since our data mainly comes from chromosome 22. And in order to do so, we will click here on this button, filter data on any column using simple expression or search for this tool. And we apply it to the data which we downloaded in the beginning. So this NENCFF file. already in the default mode, with following conditions, we say you check column 1, and column 1 should be equal to the string chromosome 22. And here we simply click on execute. To explain you this a bit better, what is happening now, if we would look into this peak file of Ctcf, then you see in column 1, it's always listed the chromosome name. And we simply check if this chromosome name for every line corresponds to chromosome 22. Since we want to check if ATAC-Seq peaks correspond to Ctcf binding sites, we also have to select only Ctcf binding sites which correspond to, or which lie in intergenic regions. So we have to kind of take now another file into consideration. You maybe remember we have used the tool for UCS, the Ucsc table browser. And there we downloaded basically data from Ucsc, the table browser, to get the genes for chromosome 22. And this we use now to filter for intergenic binding sites for Ctcf. So we click here on this tool, BED tools, intersect intervals, find overlapping intervals in various ways. And then we select here on file A to intersect with file B, the data we have, or the result file we have now generated. So this filter on data 1. And under file B to intersect with file A, we select this chromosome 22 genes file, which we have downloaded. And then we say, write the original entry in A for each overlap. So here, what should be written to the output file? And we select this -WA option. Furthermore, the required overlap, we leave in the default 1 base pair. And I will now search for this. It's right underneath it. It says here, report only those alignments that do not overlap with files. So click here on yes. Again, so we only want to have binding sites which are intergenic, so not overlapping, so not in genes itself. And therefore, we activate this option. And that's already it, and we execute the tool. That shouldn't take long. And in the meantime, we will generate another file that we need for the visualization tool. We have in Max, or we got from Max2 a BD graph file which says BD graph treatment. So if you look into the history, you find it. Where do I find it? Here, Max2 call peak, and in brackets it says BD graph treatment. And we have to make or convert this BD graph into a BigWig file, which is a binary form of a BD graph file. And it is needed for our tools, which we will use later on. So click here, Wig, BD graph to BigWig. And we apply this tool to the treatment. And simply click here on execute. And hopefully this will be done shortly. Oh, it actually failed. I have to check what happened. Let's take a short break and I come back describing what I have done wrong. So what did I do wrong? I checked, and technically I did not something wrong with applying the tool. However, what I did wrong is how I downloaded one of the data files, especially the chromosome 22 genes, the annotation. I hope you followed the training material and not my instructions right in the beginning. So if you followed my instructions, there in the training material, it's written when you apply the Ucsc main table browser to get the chromosome 22 genes. I said I used the I took the database gen code version 36. However, you have to take the database gen code version 37 here. And also the table is the basic table. So if you also run into a problem, into an error, then this is probably the case for you as well. Please go back, down, follow the training material, select all the options which are written here. Then apply again this cut columns from a table tool and rename your file into chromosome 22 genes. With that, it should work. And therefore, we come back to where we left. So I applied the B2 tools intersect, and I applied this conversion of BD graph to BigBig on the treatment file, so for our coverage. In the training materials now written, so to better have an overview, we renamed now this BD tools intersect file. So this is basically, these are our intergenic Ctcf peaks from chromosome 22. And the BigFig results we simply name next to BigFig. So maybe I was too fast. So simply copy this text here for the BD tools intersect file, and copy this text here for the BigFig file. And then to rename again, so click here on edit attributes, and then paste the name here and click on save. With that, we should have everything. However, we have to first compute now for the visualization tool to inspect the transcription start sites and matrix. And this matrix here, so this compute matrix, considers or creates as a matrix for the transcription start sites to see what we would expect that the coverage should be increased for our ATAC-Seq data around transcription start sites. And therefore, click here on Compute Matrix or search the tool in the search tool field, and select under Select Regions to plot the chromosome 22 genes. Sample order matters, no. The score file is the max2 BigFig file, which we generated. And under Compute Matrix has two main output options. We select, instead of scale regions, we select the reference point. And already in default, we say beginning of a region. So this we leave. And we also leave in a default distance upstream and distance downstream to 1,000. So we pick a big window, so of 1,000 bases left and right to the transcription start sites to look at the coverage. The only thing which we now have to also select is under Show Advanced Options, yes, and convert missing values to 0, yes. With that, we are good to go, and we simply execute the tool. I hope this does not take too long. No, I'm staying in the queue. Let's see. I hope it will be resolved quickly. If not, then we take a short break here, and I come back in a moment. So now the compute matrix should be completed, and we continue now to plot the heat map. So we click here on plot heat map, We select the matrix file from the computation metrics tool, what the result which we generated. And that's it. We simply click on execute. And this time I hope that it doesn't take long and that I don't stay in the queue for too long. I also have to do rake again. Or we do already in the meantime. Yeah, let's do this in the meantime. So after I will explain what we see in the plot heat map, we do another heat map, so the same thing, but we will look now at the intergenic Ctcf peaks for chromosome 22, which we also anticipate that we have an increased coverage at these sites. And for that, we will do the same tools. We apply the same tools. So we click here on compute metrics. And now instead of the chromosome 22 genes, we select the intergenic Ctcf peaks chromosome 22. We select again here on the score file the max2 bigwig. And we select now under reference point, compute metrics. We select here the center of the region, at the beginning of the region. So the reference point for the plotting center of the region. Again, left and right, we leave 1,000 bases. And we do the same thing, show advanced options, yes, and convert missing values to 0. And we click then on execute. Okay, now let's come to the results of our heat map, because I saw we are now finished with the heat map, the first one, and we simply inspect this by clicking on the view data button. And we see, so on this upper plot shows you the average coverage, the blue line. On the y-axis, the coverage. On the x-axis, the distance to the transcription start size here in the center. And the heat map shows you on the x-axis, on the y-axis, so to say, all the genes which we had in our annotation file. On the y-axis, on the x-axis, the distance again to the transcription start sites. So left and right, 1,000 bases. And then you have from red to blue a cover, so the scaling of the coverage. So blue means high coverage, red means no coverage. And you can see that around the transcription start sites, what we expected, we have an increased coverage, which is actually shifted to the left, which we also would expect, because on the left we have more upstream, the upstream regions. And the upstream regions are normally quite often correspond to open chromatin regions. So here it's a bit left shifted. Okay. Now the other matrix is generated and finished. And with that, we can apply again the plot heat map tool for our second result file here. So we click here on plot heat map. And we select now this compute metrics result, so the 58 for me here. And we will change a bit the design of the plot. So under advanced options, say yes here. And then color map. So I have to search for this color. Exactly. So on here, color map to use for each sample. We click on insert color map to use for each sample, and then we select blue. You remember we had a red to blue scaling. Now I change it to blue scaling to show you that you can change the appearance of this plot. Then we also label the x-axis a bit differently. So simply copy this text here and then go to the x-axis label And you may remember from the other plot, it was distance from the transcription start site. However, we have not now inspected the transcription start sites. We inspected the intergenic peaks or Ctcf peaks. Therefore, we name this distance from the peak center in basis. And also the y-axis we relabel. So under the y-axis label for the top panel, we say here Ctcf peaks, since we inspected Ctcf peaks. And reference point label, we say, so there's also reference point label. We say transcription start sites. We say peak center, and we say for labels for the regions plotted in the heat map. So here we say Ctcf peaks. So I'm just checking again if I selected all the right options. And then we only have to go now to this option here. Did you compute the matrix with more than one groups of peak regions? Yes, I use multiple groups of regions. And then we click on execute. So I just wait a short moment. Or maybe it's doesn't take too long. Ah, it's already computing, that's good. And then it should be finished in a few seconds. So I mean, you can also skip to the point now where the heat map is finished. And it is finished now. So if you click now on the View Data button, then you see, again, the top panel is the average coverage. And you already get the idea that, yes, what we anticipated, the coverage is increased around the peak centers of Ctcf intergenic peaks. And it's also not now left shifted, since we don't have transcription start sites, we have intergenic peak sites of Ctcf. So it's technically really centered around this peak center. And you can also see that we relay the labels which we changed for the x-axis, the y-axis, and also here the coloring for the heat map. So also here the labels, peak center and Ctcf peaks here for the y-axis, and the coloring is changed now to just a blue coloring scheme. Okay, with that we do a short break and I come back in a moment. So let's come to the last point of this tutorial. And we will now inspect some of the results in an integrated genome browser in Galaxy. You may know already genome browsers like IGF or the Ucsc browser. However, Galaxy also supports several genome browsers like Genome Tracks to make it for you a bit more handy to inspect certain regions of your data right in Galaxy itself. And what we now want to inspect is kind of what we already did or slightly did with the plot heat map. We first of all want to check, do some of the peaks correspond to transcription start sites or intergenic Ctcf sites. On the other hand, we also want to see if we could find peaks which do not correspond to either one of them, and therefore mark other regulatory elements that might be of interest. So this is something which you typically do in ATAC-Seq, of course, to see what kind of regulatory elements you have in your data and how do they correspond to certain transcription factors, transcription subsets, and which are kind of novel and maybe of interest for further analysis. So let's take this tool here, Genome Tracks. So click on it, and we will now create our tracks for a certain window in there. So stay with me. There are a lot of options which we have to change, but at the end, it's a compact visualization. So first of all, let's copy these coordinates here and go to region of the genome to limit the operation and paste these coordinates in the text field. And then we start to formulate our tracks. So the first track is already, so we already have a default first track here. And for that, we simply select under choose style of the track, a big track. And the track files, big format, we select here the max2 big file. And the plot title, we copy here from the training material. So on the plot title, we paste the text here. And then color of the track is now up to you. And the training material is not specified. So let's see, I will select now yellow. And minimum value is 0 and maximum value is 5. And show visualization of the data range, we show visualization of data range, yes. So minimum value means kind of the minimum coverage. So we selected our coverage track, so the minimum value this coverage track has, and the maximum value. From this plot heat map, you know, the average is around about 5. So therefore, we selected here the maximum value of 5. And show visualization of the data range just means, yeah, we showed the visualization. With that, we have already our first track. And now we click here on Insert Include Tracks in your plot. So we select, or we include now a second one. And for the second one, we choose style of the track. We select narrow peak track. The plot title we can copy from the training material. These are the peaks from max2. And we select therefore from max2 the call peak on data 30 narrow peaks. So these are the peaks the peak region, so to say. And then color of the track, you can select again on your own. I take here this color and display to use. So we scroll down here, display to use. We want to display it as a box. So just draw a box for this region. And plot labels. Plot labels, name, p-value, q-value. No, we don't really have names there, so we don't want to plot them. So this is the second track. So your first track is the coverage track, second track are the peaks. Now we want a third track. And for the third track, we will take the gene track, BED track. We copy the name genes. So we want to see the chromosome 22 genes now, and therefore we select on our track files the chromosome 22 genes. Again, the color is up to you. Select now here, stay with black. And height of the plot is 5. And plot labels, yes, this time. So the height is basically just simply the height of these boxes, which we will draw. And the plot labels we want to have, so we want to see the gene names. So therefore, yes, here. And plot put all labels inside the plotted region, yes, and allowed to put labels in the right margin, yes. So this is basically taking care of the labeling. Okay, that's it for this track. Now comes the fourth one. So we have coverage, peaks of our ATAC-Seq peaks, and we have now our genes in there. Now we want another narrow peak track. So I include the fourth track, so as I mentioned, and we want to have the Ctcf peaks, as I mentioned. So plot title Ctcf peaks. And we have this here, this intergenic Ctcf peaks from chromosome 22. Ah, no, pardon. Not these, not this file. We want all the peaks. So not only the intergenic, we want all the peaks. So therefore we select this ENCFF BD file. And then, yeah, track color, again, up to you. We'll select here now. I would select kind of a red. And under display to use, box, draw box. And plot labels, again, no, we don't really have labels there. And these are our fourth track. The fifth one, which is now missing, is simply an x-axis. And therefore, we insert another track, and there must be something on the choose star of the track, which is named x-axis. And that's it. And we simply execute. You can also change the image output format from a PNG to an SVG, but let's stay with a PNG and simply click on execute. And I hope this doesn't take too long. Okay, I think stay in the queue again. So let's take a short break here. So I hope you now finished the Pi Genome Tracks, and I did it again. So that's why the change of background and change of shirt. Because I saw later on that I did something wrong and I had to correct it. So as you can see, so this is original pictures. So this is how it would look like if you really followed how I did it. And the problem already, so I did it again because the color here is maybe was not a wise choice. And also the height is at 5. So I said, okay, we know that kind of the maximum is 5, the coverage is 5, but that's not true. If you follow the training material, then you see instead of the maximum value in the coverage track of 5, so for the first track here, so I selected here maximum value of 5. In the training material, it's actually written, and I show it to you, that you have to have no maximum value and the height is 5. That's the change. And so this looks now like this. So not that you get confused why your plot looks a bit different to the training material. And what you can see here now is all the tracks which we have selected. So the first track is the coverage track for our ATAC-Seq data. The second track are the peaks from our ATAC-Seq, called with max2. The third track are the genes, which we see in this window. I also mentioned beforehand that Pi Genome Tracks is kind of a genome browser. I have to correct myself there also. That's not true. We see here a certain window, so it's not a interactive tool, so to say. So you cannot simply move around here. It's just this window. So that's why we see this certain window here, and we select this certain window. However, Galaxy has some genome browsers available, which you can search for. And the fourth track are the Ctcf peaks. And on the x-axis, so the last track is the coordinates, so to say. And we can see that we have here in this window 4 peaks, 4 ATAC-Seq peaks, which corresponds to these big spikes here in our coverage track. And we see that we have 2 genes, so RAC2 and SSTR3, with 6 transcripts, 4 transcripts for the first gene here and 2 transcripts for the second one. And more or less, we have 3 transcription start sites. So this transcription start site here and these 2 here. And we can see that one ATAC-Seq peak more or less corresponds to the transcription start site of RAC2. Furthermore, we see that we have several Ctcf peaks here. It's not clearly visible, but the last peak of our ATAC-Seq data here corresponds to one of the Ctcf peaks. So with that, we confirmed that some ATAC-Seq peaks correspond to, again, to transcription start sites, some to known transcription factors like Ctcf. And then we have 2 peaks in the middle here, which does not correspond to any of them. What you would do now is to figure out what they correspond to, what kind of regulatory element is that. So you will look for another maybe transcription factor regions for that, maybe histone marks or methylation sites, something like that to confirm that this is a valid open chromatin region. And or closed chromatin region to deconfirm it, so to say it's actually a false positive here. And so this goes then into the further peak analysis which you would do. So with that, you see it's very important to look into your data again within Genome browser to confirm some of the sites and to see to answer your biological question. With that, we come to an end. This was the last part of the ATAC-Seq training, and I want to summarize. So at the end, you should know by now what ATAC-Seq is and how you get annotation data, first of all, for the later data analysis. For example, by the Ucsc table browser, you should know now how to, first of all, check the raw data quality with Fastqc. Then you should be able to do a quality and adapter trimming with ctDapt, followed by a mapping with Pertile 2. Furthermore, also checking there the mapping quality and some quality checkups with, again, Fastqc. Then you should be able to do specific filterings for ATAC-Seq data, like filtering out reads that are properly paired or correspond to reads that map to the mitochondrial chromosome, which are not of interest. Then you should be also able to filter out Pcr duplicates, which you then also check with a quality control. You should also be able to check this insert size pattern of ATAC-Seq data, which is a very important checkup for it. And then you should be able to do the identification of open chromatin regions with MAX2. And then you also should be able to further analyze these peaks, these identified regions, with Genome Tracks and DeepTools. So DeepTools is this heat map for the transcription start sites and the intergenic Ctcf peaks. Of course, there's a lot of other things you can do. We covered here kind of the basic for ATAC-Seq data, but there is still further analysis you would do. And if you have further questions regarding this training, then please let us know. During the training itself, maybe there's a Slack channel available, so write a message there asking questions, or try to find us. So me or Lucille are also available via email. So you can ask us some questions regarding ATAC-Seq data analysis. Okay, thank you very much for your attention. And I wish you with that a good day, a good week, and I hope I could help you to analyze your ATAC-Seq data. Okay, bye-bye.